Now, we'll be investigating the effect of temperature on transpiration using a bubble pottometer. For this experiment, we'll need a water basin, a scalpel or a sharp pair of scissors, the pottometer itself, Vaseline or oil to help create airtight seals, and a paper towel for drying the plant stem before setup. We'll also be using a timer to measure the duration of each trial, a lamp to provide constant light, a ruler for measuring distances, a thermometer to monitor temperature, an electric fan set to keep air movement steady, and an anemometer to check that airflow remains consistent. On our diagram of the bubble pottometer, you'll see the key parts labeled, the fan set, the term, transpiration, to indicate the process we're measuring, the thermometer, the transpiring plant with its cut chute, and the timer. You'll also notice the anemometer, the rubber bung securing the plant into the reservoir, the capillary tube containing the air bubble, and the lamp providing constant light. The volume scale on the tube is used to measure how far the bubble moves, and at the end of the tube is a beaker of water where it rests. By using this setup, we can carefully control the variables, change the temperature, and measure how it affects the rate of transpiration in our plant sample. For this experiment, we'll need a water basin, a scalpel or a sharp pair of scissors, the pottometer itself, Vaseline or oil to help create airtight seals, and a paper towel for drying the plant stem before setup. We'll also be using a timer to measure the duration of each trial, a lamp to provide constant light, a ruler for measuring distances, a thermometer to monitor temperature, an electric fan set to keep air movement steady, and an anemometer to check that airflow remains consistent. So, for this experiment, the experiment involves first cutting the plant chute underwater to make sure no air enters the xylem. Then, we assemble the bubble pottometer while still underwater, sealing all the joints with Vaseline or oil to prevent any leaks. Once assembled, we place the pottometer on the workbench with the tip of the capillary tube in water, and we dry the plant's leaves so that no excess water blocks the stomator. We then introduce a single air bubble into the capillary tube and return the tube end to the water. Next, we place the setup in rooms with different, stable, and controlled temperatures. Using a thermometer, we record the temperature in each room. Over a fixed period of time, around 30 minutes, we observe the air bubble's movement in the capillary tube and measure how far it travels. That distance tells us the rate of transpiration. We repeat the experiment at each temperature at least twice to make sure the results are reliable. And throughout the whole process, we keep all other conditions, like light intensity and wind speed, constant, so temperature is the only factor affecting transpiration. So we'll observe that as the plant transpires and loses water through its leaves, the air bubble in the pottometer moves along the capillary tube. That movement represents the amount of water the plant has taken up. In each temperature condition, we'll carefully watch how far the bubble moves over a set period of time, recording the distance at regular intervals. This gives us a clear, measurable way to determine the rate of transpiration. By comparing how far the bubble travels at different temperatures, we'll be able to see how temperature affects water loss. If the bubble moves faster, it means the transpiration rate is higher, more water is being drawn up through the xylem and lost from the leaves. The distance the air bubble travels in the pottometer provides a direct measure of the plant's transpiration rate at different temperatures. In general, higher temperatures result in faster bubble movement, indicating an increased rate of transpiration. This occurs because elevated temperatures speed up the evaporation of water from the leaves, prompting the plant to draw more water through the xylem to replace what is lost. Conversely, at lower temperatures, the bubble moves more slowly, reflecting a reduced transpiration rate. Cooler conditions slow evaporation from the leaf surface, leading to less water loss and slower water uptake. These variations in bubble movement clearly illustrate how temperature influences the plant's water loss through transpiration. So, temperature has a clear and significant impact on the rate of transpiration in plants. In warmer conditions, transpiration rates generally increase because higher temperatures accelerate the evaporation of water from the leaf surfaces. This creates a greater demand for water to be drawn up through the plant's vascular system, resulting in increased water uptake and a higher overall transpiration rate. In contrast, cooler temperatures slow down evaporation, reducing water loss from the leaves and leading to a lower rate of transpiration. Overall, the experiment demonstrates a direct link between environmental temperature and plant water loss, highlighting how changes in temperature can influence the efficiency of transpiration.